So who gave us this model of DNA, this double helical structure model of DNA? And here comes into play James Watson, Francis Crick, and Rosalind Franklin, which I'll discuss in a bit. They're biologists who discovered the double helix structure of DNA. They won a Nobel Prize in medicine in 1962 for their work. They decided to use this model-based approach to basically discover the molecular structure of DNA. So here we can kind of see this molecular model that they're using in order to decide if this is going to be how DNA is structured in our own bodies, in our cells. And they figured this was important to discern the molecular structure of it because they are really wanting to discover that geometry that would be central to discovering how DNA could transmit genetic information from parent to daughter cells. So like they already had that background on Mendelian genetics and that idea of heredity. And so they're wondering on a molecular level, how is this heredity going to be passed down from parent to daughter cells in the form of gametes? So Watson and Crick basically used this new information that they built on previous scientists to construct this double-stranded helix with nitrogen bases pairing A to T, C to G. So let me show you an image where they figured, okay, I have a sugar phosphate backbone side and I have nitrogenous bases kind of hanging off in the center. And there's a pairing effect that goes on with another strand. So it's partnered strand that's going to also have those bases hanging off to the side. And in the middle here, the bases will bond to each other. So they use this kind of wire model to show that. Building on with Rosalind Franklin's X-ray crystallography images. So points out down here that Franklin used X-rays to generate some of the key data that was important to discovering now this kind of twisted helical model. So here we can see an image, and in that image there's an X formed in the center in her X-rays. That X is extremely important because we wouldn't see this X forming if DNA was so structured like a ladder, like Watson and Crick originally thought. So they built on that idea and like, okay, what if we take that ladder and twist it so the strands, that sugar phosphate backbone, are now twisted, and you still have that hydrogen bonding occurring between the different nitrogenous bases that hang off to the side. And we can actually see that kind of X pattern being shown by those more dense sugar phosphate backbone. And that's what Franklin was showing in her X-rays. So Watson and Crick also used her research. Unfortunately, didn't credit her with that research. Later on, the Academy did Franklin's research, and they built that model to show that twisting helical structure of DNA. Okay, so this is a beautiful molecular explanation showing us how genes can be passed down from parent to daughter cell, where those genes are located in the form of DNA.